Welcome to the cell cycle video. In today's video we're going to discuss the cell cycle, the different phases a cell goes through, and the importance of each of those phases. So we're going to begin, again, just to recap, talking about the cell, and it's important to note those structures and don't forget about that, and going back to the cell theory, what it states, and that we're really emphasizing and trying to understand how cells arise from a pre-existing cell. And going back even further and talking about the functions of why a cell would need to undergo cell division as a whole, there are three major reasons why a cell would want to undergo cell division. It's for growth and development, for repair and maintenance, and also for reproduction. So what I'd next like to talk about is the cell cycle. So interphase is characterized by G1, S, and G2. So this phase is called interphase. And the second phase of the cell cycle is called mitosis. And mitosis begins right at the end of, so right here at this line, and that's where it begins, and it ends right here. So it's the shortest phase of the cell cycle and the most exciting phase of the cell cycle, because this is where we begin to see the cells dividing into two. So there are some cells that actually exit the cell cycle and they go into a resting phase, which is a G0 phase. So in the G0 or G0 phase, they are not actively growing, they're not actively synthesizing their DNA or undergoing cell division. And so there are some cells that exit the cell cycle and go into the G0. So those are the phases of the cell cycle. Interphase is characterized by G1, which stands for gap 1. And what happens in G1 is we see that the cells begin to grow. So there's a lot of growth. It's actually where most of the growth takes place inside in a cell, is in the G1 phase. We see the duplication of many organelles, and we see expansion of the cytoplasm as well. So cells grow and they're getting ready for cell division. So they're trying to prepare for cell division in the G1 stage. And this can take a very long time, depending on the cell type. The next phase of interphase is the S phase. So S stands for synthesis, and it's synthesis of DNA. So in this phase, the cell is duplicating its DNA, and we want to make sure that our cells have the same amount of DNA, and in order to ensure that, cells have to undergo duplication process. So this is a very important stage because we see DNA replicating or duplicating, and there is no growth taking place at this phase. So this is only for DNA duplication, and there's a lot of regulation proteins that make sure that accuracy of the DNA when it's being replicated is with high fidelity, which means that it's very accurate. The next phase of the cell cycle is the G2 phase. So in the G2 phase stands again for gap. I like to think of it, by the way, as growth, and I like to tell students that you can think of G as for growth, but it really stands for gap. So we have the G1, which is the first growth phase, then there's synthesis, so there's no growth here, and then the G2, which is the second growth phase. So there's 3 phases total, but 2 just for growth, and one of those phases is for synthesis of DNA. So in the G2 phase, cells will finish up growing, and they're preparing to enter mitosis. And in mitosis, right here, this is the shortest phase. This is where we see prophase, and we see metaphase, anaphase, and telophase take place. Mitosis is arguably the most exciting phase of the cell cycle, but I am biased. This part of the cell cycle is quite amazing. Okay, so now what I'd like to do next. So I have an animation to show you, and in this animation, this cell is going to divide eventually, and right before it divides, though, it's going to show here in this animation the different phases that a cell will go through before it divides. The cell here is showing its DNA, and the DNA will become duplicated, as you see there. So as it's finished duplicating, it'll enter the G2 phase, and then it's going to enter mitosis. And in mitosis, we see the nuclear envelope breaking down, we see the centrioles moving to the opposite sides of the pole, the chromosomes moving to the center because those microtubules are pulling it, and then finally those chromosomes get pulled to the opposite side of the cell, the nuclear envelope will reform after that. And that's the entire cycle of the cell. All right, so some of the things that I wanted to emphasize, it's really important to emphasize, is why is it important to study the cell? Well, if we study the cell, then we can understand how diseases work. And a particular disease that comes to mind immediately is cancer. So cancer is a dangerous disease to have in your system because once cancer takes hold, it's very hard for your body to regulate those cancer cells because they technically are self cells, and so your immune system will have a hard time fighting that off because it thinks that it's a normal cell. It has no idea that it's cancerous. And I want to show this animation that describes how a cell is so highly regulated. So now we see in this animation something called a checkpoint. You don't have to know how many checkpoints exist inside the cell. There are 3, by the way, 3 major checkpoints. But I want you to understand that there are these signals, and they act as like a go signal inside the cell. And these signals are essential for making sure that the cell can duplicate its DNA and make sure that the chromosomes separate evenly, and also make sure that there aren't any problems inside the DNA as well. Because sometimes what can happen is cancer can arise due to high levels of energy, such as UV radiation. And that UV radiation can damage DNA. It can damage DNA in certain regions where it can destroy genes that are important. Sometimes mutations can take place, and a cell that normally would die because it's malfunctioning might not die anymore because of a particular mutation that took place. And so that cell can become malignant in that case. The reason why a cell is malignant is because the cell divides uncontrollably. There's no regulation on the cell itself. It'll just keep dividing and dividing. There aren't any checkpoints when the DNA inside a cell has mutated. And so when the DNA inside a cell is mutated due to high levels of perhaps radiation, it can also be due to viruses. Viruses have been known to cause cancer. And there are other toxins that can cause cancer, such as smoking can cause cancer. And these events will mutate the DNA and cause the DNA to become malignant and then prevent it from obeying certain signals inside the cell. So normal cells would rather die than live on and become malignant. And so all of our cells work together, and whenever there's a problem, it'll sacrifice itself and undergo cell suicide, which is called apoptosis. And when a cell is malfunctioned, it'll undergo apoptosis so that it doesn't damage the rest of your cells in your body. Cancer, however, is a little selfish because it doesn't know how to die. And so this is an example of a cancer cell among a sea of normal cells. The cancer cell here is considered to be malignant. Sometimes you can have malignant or benign cancer cells, but a malignant cancer cell will metastasize, and that basically means it'll spread. So this would be a cancer cell which will eventually keep replicating, and it won't stop replicating. It'll continue to replicate, and it'll continue to divide until finally it's starting to push the normal cells out, and this will eventually spread into the bloodstream. And when it spreads into the bloodstream, this is considered to be a metastasized cancer, and this can start infecting, or I should say invading, other organs inside your body and spread. So whenever you hear about the cancer spreading, this is exactly what they're referring to, that the cancer will get into the bloodstream and then spread to the rest of your body. So this is quite a fascinating topic, because just like how an odometer works inside your car that tells you how far you've driven, a cell has an equivalent odometer that tells it how many times it's divided. So of course it doesn't have an odometer, but it has a memory. So cells actually have a memory, and they know how many times they've divided in the past. And as it turns out, a cell has a limit to how many times it can divide before it undergoes apoptosis or cell suicide. This is a quite fascinating topic, and it's one that's only been recently discovered in the past 50 years. And cancer, however, doesn't have an odometer. There is no clock that will tell the cell, say, okay, you've multiplied over 52 times, because that's actually the limit is about 52 cell cycles, and when the cancer cell reaches 52, it doesn't register. It will just keep dividing over and over again, and every time it divides, it becomes more and more malicious, and it becomes more and more invasive because there's most likely going to be more and more damage every time it's dividing. Well, that is summing up the cell cycle and kind of stringing together some of the important factors of the cell cycle, such as cancer and regulating cell growth. So I hope that was helpful, and keep studying.